Very good evening. Very good evening, Moksh Sharma. Good evening. Good to see all of you. Seshagopalan, Moks, Smart Single, Smart and Single, Lake Paul. Right. Glad to see you. So, anyone who gave the exam here? So, absolutely, Seshagopalan and Moksh Sharma, they didn't give the exam. Smart and Single. So, in which year are you? Lake Paul. So, we have one first year student, one third year student. Right. So let's discuss some of the questions which have been asked in INICET exam. Right. Look at the question. So you know that I put this question in our Telegram group also. So this question was there in the Telegram group. Similar question. It's not that I put exactly the same question, right? So let me give you the poll. Excellent, Rashmi Moks. So the same thing I explained in our Telegram group also. Still people are choosing the wrong answer. I'm just having a problem with my writing pad. I'll try to correct this. Excuse me for this trouble. Yeah, it's now solved. So guys, you remember I was telling you that telomerase is a reverse transcriptase. Telomerase is a reverse transcriptase. Why telomerase is a reverse transcriptase? Because it carries its own RNA template. So you see, this is the telomere. So this is the telomere, right? So 3 prime, this will be 5 prime. So what is the problem? Here the RNA primer is gone in the lagging strand, so there is a gap. So telomerase will come and it will elongate the 3 prime ending strand. So how it is doing? The telomerase has RNA template. Seshagopalan, Mox, you remember? We have already discussed this, right? So the RNA template is carried. It's carried by the telomerase. Telomerase is a ribonucleoprotein, but it is not a ribozyme. Okay, this telomerase is not a ribozyme. So telomere is repetitive DNA. You know, centromere, telomere. So they are all repetitive DNA. So they have a particular hexanucleotide structure which is repeating. So this telomerase, it carries a sequence which is complementary to this hexanucleotide structure. Okay, so it carries a complementary sequence of this hexanucleotide sequence. Is this clear, everyone, now? So telomerase is a reverse transcriptase. It is found only in eukaryotes. Yes, because telomerase helps to solve the problem called end replication problem. You find end replication problem only in eukaryotes, because in prokaryotes the chromosome is what? Circular. So in circular chromosomes there is no end, so there is no need to have the end replication problem. Okay, so telomerase, it maintain chromosome length. That is absolutely correct. Why? Because when the this lagging strand primer is removed, right? So when the lagging strand primer is removed, so there is a gap. So this gap with successive replication. So Hayflick's limit, Seshagopalan, you must be remembering. So we have discussed Hayflick's limit. So Hayflick's limit, so yeah, so it depends on the cells also, Seshagopalan. So it depends on the organism. So what is Hayflick's limit? It tells that the number of times an adult somatic cells can divide. Okay, so number of times adult somatic. Yeah, option D is different. Option D is different. So if you can, please tell me the option D. So option D is different. I know that. So that is so the thing is, many students, so that's what I have to thank the students who recalled and told me in this short gap. So recalling the questions is actually a tremendous job. Okay, thank you, Dr. Sushant. So DNA fidelity increase by telomerase. So DNA fidelity. So what is the reason for fidelity? Tell me. So fidelity is by exonuclease activity, right? So fidelity of DNA replication is by 3' to 5' exonuclease activity of DNA polymerase. That's what fidelity. So what is fidelity? It is the faithful copying. So the same sequence is there in Seshagopalan. The same sequence has to be come in the daughter strand also. That is called fidelity. Okay. So fidelity is like commitment, you say. So same sequence coming, that is called infidelity. Clear? So fidelity of DNA replication is due to the proofreading activity. So proofreading activity is responsible for fidelity. So thank you, Dr. Sushant, for telling the correct option. So very soon from Unacademy end, we will release a PDF. So there we will try to give the correct options and all. Okay. So thank you. Next one. So this is again option D. That's what in option D. Option D is not clear. So okay. So most of the things, 3 options were told by the students correctly. So if you know the 4th option, please tell me. Okay, it is thalassemia. Excellent, Dr. Sushant. So knuckle pigmentation. So what is the other condition in which you see knuckle pigmentation? Tell me. Is there any other condition you will see knuckle pigmentation? So what you are seeing is the knuckle pigmentation. So which is the other condition in which you find knuckle pigmentation? So one is B12 deficiency, and also B12 deficiency that leads to megaloblastic anemia. Moksh Sharma. Any other condition? Addison's disease, right? So you find knuckle pigmentation in Addison's disease also. Okay. So this is a simple question. So okay, this is medicine, biochemistry. Okay, so both subjects you can put right. Next one. So this is an interesting question. So there are students, like first year students are also there. So I will give some time for them to do the calculation and all. So I'm increasing the time. I'm giving the poll. So let me A, B, C, D. Let me give the options. I'm correcting the options. Okay, A, B, C, D. Jaydas, welcome. Jonas Sharma, Mukesh Gupta, Neeraja, Alison, Aryu, Sai, Samudra, Twinkle, Shreya, Dr. Sushant, Bismillah, Sujina, Tamil Dr. Anuradha, Kritika, Deepak, Dr. Adar, Kemeshwar, nice, Namrata, Rashmi, Pragya, Arjun. Okay. Nadia, welcome. Right. So this question is based on this knowledge, something called catalytic efficiency. Catalytic efficiency. So what is catalytic efficiency? How good an enzyme is. So that catalytic efficiency is what? It should be Km by Kcat or Kcat by Km. Tell me. So it will be what? Kcat divided by Km. Clear, Moksh Sharma? You forgot. Hmm? So it's Kcat divided by Km. Why? Let me tell you the logic. It's very simple. Hi Salim, welcome. So catalytic efficiency, the formula is Kcat by Km. See, Km is inversely proportional to affinity, right? So inverse ones should come in the down. Clear? So inverse ones 1 by Km. Clear? So Kcat is directly proportional to the activity, how fast the enzyme, right? So now look at this. So they have given, how nicely they have given. See, look at this. So Km micromole. So the in exam, I think M, small m, capital M would have come. Dr. Sushant. So how was this? It was small m, capital M, or they have given micromole? Hmm? Hi Salim, I am good. So it's after a long time I'm seeing you. How are you doing now? Okay, so they have given micromole, right? Good. Right. So it will be small m, capital M if they are writing in a short form. So whatever it is, you convert the nanomole also into micromole. So finally you convert nanomole into micromole. So how much you will get? Tell me. 200 nanomole. Oh, Salim. Glad. All the very best. Yeah. Today is paper discussion. Hmm? We are discussing today's paper only. Hmm? So convert nanomole to micromole. It is 0.2. Excellent. Hmm? So if you do Kcat by Km for everything, so ultimately here what will you get? Hmm? So everything if you do, ultimately the highest one you will get here, it will, you will get 100. Okay. So that is why this is the correct answer. Okay. Hmm? Clear na? No confusion? Right. What will you get for option B? Hmm? For option B, what will you get? 50. Right. Hmm? 50 divided by 0.2 you do. So do the calculation. It's very simple. Okay. So we are getting 250 there. So then I think then there is wrong recall this. Okay. So here some option is not there. Okay. So you all chose C correctly, right? So here we have to change the value. Okay. So the recall was, this was the correct one. So here things are different. Okay. So change accordingly. Hmm? Right. So please change the option B. So we will release a final PDF. There we will correct everything. Now answer this one. In vitamin B12 deficiency, which of the following will be involved? Okay. Yeah, the concept is same, but finally we have to give the correct one. Okay. So guys, we will give the final PDF. Okay. So here we are just going to discuss the concept. We will release the final PDF. Okay. So take it as that as a final. Excellent. Salim, Deepak, Shreya. Good. Many of you are answering nicely. Hi Sneha. Again, this is a simple question. And in B12 there are 3 questions, right? So B12 is such an important topic. So there are 3 questions: hypersegmented neutrophil, image based question, this question, biochemistry question, right? Vitamin B12 is such important. Which one was there? So the question was level, they asked. Like which one, Kritika? Increase, they asked. Level increased. They have given the enzymatic activity or something else. Okay, so let's discuss the concept first. So in vitamin B12 deficiency, okay, but they ask increased. I see. Hmm? So what's happening in B12 deficiency? Methionine synthase and methylmalonyl CoA mutase. So this is what methylmalonyl CoA mutase. So both are decreased. So there are just 2 B12 dependent enzymes, right? So the just B12 dependent enzymes, 2 B12 dependent enzymes are there. So methionine synthase and methylmalonyl CoA mutase. So which level of which will increase? Which metabolite will increase? There will be increase in methyl malonyl CoA. Is this clear? Methyl malonyl CoA will increase. So that is why you will have methyl malonic aciduria. You will have methyl malonic aciduria. So what about homocysteine to? Okay. So Dr. Sushant is saying, I think they asked which reaction not take place in B12 deficiency. We will verify that. Okay. Don't worry. We will verify that. So another one is what? Homocysteine to methionine. So many people are thinking that in homocysteine to methionine, what is involved? Homocysteine to methionine, which one is involved? Tell me. Conversion of homocysteine to methionine. What do you need? Methionine synthase only, right? So here B12, B9, both are involved. Folic acid as well as B12. So here homocysteine to cystathionine. Homocysteine to cystathionine. That which one is involved? B6 is involved. So somewhat homocysteine level will also increase. So if they ask, I don't know what they asked. So if they ask increase in level, just they give metabolite. What are the levels will increase? The level of homocysteine will be high, methyl malonyl CoA level will high. If they ask which of the enzyme activity will decrease, then it will be methionine synthase and methyl malonyl CoA mutase. So the methyl malonyl CoA mutase is nothing but an isomerase. Okay, so mutases are isomerases. Right. Let's see this one: steps of PCR reaction, polymerase chain reaction. I think this is pretty straightforward for many of you. I don't find difficulty in this question. So this is what I am saying. You focus on the concept. Many times what happens na, the recall is not proper. You can see clearly, right? Students, they don't recall properly. See, what we can discuss is from the recall given by the students only, right? You don't get official questions anyway. So that is why, don't, whenever you see the past question, if there is something like fishy and all, don't worry. That is not the exact recall. So you always try to focus on the correct concepts. Okay, so Many students, what they got confused is between the hybridization and annealing, right? So they are like interchangeable, hybridization, annealing. So don't worry. Let's see how this is happening. So this DNA. So in the PCR, what will be the first step? First step, we have to separate the two strands of DNA, right? Heat denaturation. So we are going to increase the temperature to 96 degrees Celsius. Okay, 95 to 96, right? Right. Hmm? Then what's going to happen? In the mixture already primer is there, right? So primer is going to anneal. So we usually use the term primer annealing, right? So forward primer and reverse primer. So they are going to anneal. Then the Taq polymerase. The Taq polymerase. So now we are going to change the temperature into 72. So the temperature will be different, right? So everything. So annealing temperature will be different. Hmm? So the optimum temperature of uh, Taq polymerase is different, right? So annealing temperature should be lower than the melting temperature. Hi Nitish, good evening. So it is denaturation, annealing, then elongation. You are not too late, don't worry. So we have questions, don't worry. Clear? So denaturation, annealing. So you are understanding the term annealing, right? So I want you to understand the term annealing. Annealing means the primers, they go and make base pairing. Hi Praveen Kumar, good evening, welcome. So that is called annealing. Are you understand? So hybridization you can say, but mostly we use probe hybridization. You remember? So in fish and all, so that there we use mostly that term as that. Both are interchangeable only. Fish, right? Fluorescent in situ hybridization. So probe is bigger than your primer mostly, right? Is this clear? Let's go to the next question. Salim, we can avoid specific questions like this in the public forum, right? Hmm, right. So compare the normality of 2 millimolar HCl and 1 millimolar H2SO4. So is the question exactly same? Is this question exactly same? Right, then fine. So let me give the poll. So hereafter, before giving the poll, I will ask whether is this same or not. Nice, nice. So many of you are doing this correctly. I'm glad. So you might have, many of you might have Googled also, I know. So N is equal to M into N. Hi Dachayani, welcome. So look at this. So this is your +2 chemistry. Hmm. So N is equal to normality. Normality is your molarity into N, right? So for HCl, there is only one H, right? So there is one H. So 1 molar HCl is equal to 1 molar, sorry, 1 normal HCl. Okay. Okay. So that is what I was also thinking when I saw the question. Okay. So I seriously don't know why this question was asked. Uh, right. So I think they are testing. See, this is what. So I think they are testing your uh, previous memory and all. Okay, so I that is a thing. Yeah, yeah. This is today's exam question, Nitish. Hmm. So ultimately, see, in yeah, we had some discussion and also what I was saying is, uh, like a doctor is someone. So see, sometimes we go for intellectual discussion and also why these kind of questions comes. So it's like doctor is someone who is going to use the presence of mind, right? So ultimately it is not, knowledge is not that required, it is the presence of mind and logical skills. See, knowledge you can acquire throughout the life. Okay. So they have asked this question so that you will calculate, so it's like a maths based question, right? Okay, we will do that in our plus platform, don't worry. Hmm. So for 2 millimolar of HCl and 1 millimolar of H2SO4, they are like same because normality is molarity into 2. Okay, got it? Hmm. Right. Next one. This one is also, it must be simple for many of you. So this is based on the RQ, respiratory quotient, so which we have already discussed in many of our previous classes. Okay, they have given weight also. Good. But in the formula, there is no weight, right? So carbon dioxide released by oxygen consumed. So it will be 200 by 250. Acha. Okay, so they have a history of some patient. Good. So I don't think we will get the exact recall of that 2 line history. Hmm. Right? That's what. Varsha, don't worry. See, don't worry about the exact question. Okay. So exact, it may be flipped. Hmm. Right? So now see the calculation, right? 200 by 250. What is the 200 and 200 by 250? So you'll get 0.8. Right? Clear? Anyone want to add something? Was the values correct? Carbon dioxide 200, oxygen 250? Because the student was telling that the answer was 0.8. So for the answer to be 0.8, it should be this only. Okay, Varsha? So it cannot be reverse. Then you won't get the value of 0.8. So that is the problem. So many times what happen, I have found students, they just memorize the value 0.8 and they expect that in the next time if some similar question comes, they just select 0.8. Please don't do that. Okay. Okay, so that's what. So Varsha is disagreeing. Fine, no issues. No issues. That's what I'm saying. You just remember the formula, whatever the value come in the exam. See, uh, this is what I'm saying. So don't worry about the exact question. Okay. So RQ is CO2 released by oxygen consumed. Just know this based on whatever they ask in the exam. Okay, Varsha, Sushant, Kritika, everyone. So whatever they ask, whatever the value it is, you do it. Okay. What are you going to do with exactly by knowing exactly what they have given in the exam? Is it going to make any difference? No, right? Hmm? So you should know the concept. The concepts are repeated, not the exam questions. Okay. See, this is a concept repeat. You remember last year this same question, same typical question came, right? DNA methylation. So last year the question was something like this. So the option was like capping. Capping was the answer, right? So capping happens in RNA. It's not going to happen in the DNA. So even though it is 7-methyl guanosine, so the question was about DNA they are asked. Okay. So capping it's the RNA. Okay, so that was the last time. So this time capping was also there. Right, let's give the poll. Let's see. DNA methylation is involved in all except. Answer. Okay, wait. So change this into mismatch repair. Change this into mismatch repair. Or is this the exact option? Tell me. Is this the exact option? Or this is like same options? Hmm? Really? Okay. So methylation is involved in mismatch repair, DNA methylation. Okay. Hmm? So correct is gene silencing. This is exactly correct. Chromatin remodeling. That is exactly correct. You know, methylation, it leads to what? Heterochromatin formation, right? So it affects the chromatin remodeling. Hmm, don't know. So RNA splicing. So in RNA splicing, you have introns begin with GU, exons they end with AG. Hmm, right. Okay. Let me think about this question. Hmm, let's see. Hmm, right. This is very simple question. Okay, okay. So then fine. Then fine. Then this is fine. Okay. So then this is also not there. This is also not there. Okay. So the answer will be A and C. If it is multiple completion type, then it is fine. Okay. In base excision repair also DNA methylation is not involved. So DNA methylation is involved in what? Methyl directed mismatch repair. Is this clear, guys? Methyl directed mismatch repair. Then it is fine. So thanks for telling, Kritika. Then it is cool. Right. Right. So I think this is a exact recall. The student was able to tell correctly. Enzymes involved in gluconeogenesis. What are all the enzymes involved in gluconeogenesis? Yeah, we will give. Don't worry. So this, I think this is the easiest question maybe in the exam. Hmm? Look at this. These are all the irreversible steps of glycolysis, right? Hexokinase, phosphofructokinase, pyruvate kinase. So three kinases are enzymes which catalyze irreversible steps of glycolysis. So how can you choose that? Hmm? They're exclusive for glycolysis. Is this clear? They are not reversible. So all the reversible steps are involved in gluconeogenesis. For example, enolase. Enolase is also a glycolytic as well as gluconeogenic enzyme. So this concept is they are repeating multiple times. Let me tell you. So all reversible steps of glycolysis, they are involved in gluconeogenesis also. Is this point clear? So if there is any doubt, please ask me now itself. All the reversible steps of glycolysis are involved in gluconeogenesis also. Phosphoglycerate kinase. Phosphoglycerate kinase. So this is an enzyme of glycolysis as well as gluconeogenesis. Okay, is this clear? This is a irreversible step, right? Pyruvate kinase. How can you guys choose pyruvate kinase? Pyruvate kinase is irreversible step. You cannot reverse it with an enzyme. Sorry, pyruvate kinase is irreversible. So you cannot just reverse with a single enzyme. So okay, this enzyme. So Salim Ghazi is asking, sir, this exam paper was for what? So today there was an entrance exam, INI-CET. So this is a common entrance exam for PGA, Chandigarh, JIPMER and AIMS. Okay, so 9 AIMS and 2 colleges, so 11 colleges. So for that, this was that exam. Okay, Salim, right? Again, this option. What was the other option? Okay, Nimhans, right? Yeah. This is for postgraduate entrance exam after MBBS. Sushant, what was the option? Lipid, carbohydrate, protein, and okay, same only. Fine. Right. Then let's go for this. Thermogenic effect of food is highest for. Okay, Mox, Sheshagopalan, they are like. So thermogenic effect is nothing but SDA. So this concept also they are testing this. This is a previous question, right? So don't confuse thermogenic effect and calorific value. So that is how many people are confusing. These two are different. Calorific value is different. Thermogenic effect is different. Okay, are you getting my point? So if they're asking calorific value, then you will go for lipids. They have not asked calorific value. What they have asked? They have asked the thermogenic effect. Otherwise, diet induced thermogenesis, SDA value. Any doubt? Any doubt in this? Everyone clear this question? Very important. Diet induced. What is SDA? Okay, so someone is asking what is SDA. SDA is specific dynamic action. So it is the extra energy required for digestion and absorption of nutrient. Okay, so the whole funda is this, like this. If you take 100 grams of carbohydrate, how much do you think that, how much calories will you expect? Tell me, Allison. So if you take 100 grams of carbohydrate, how much calories you expect? You expect 400 kilocalories, right? 400 kilocalories. But when you measure, they are getting less. They are getting what? Less. So that means, so some amount is going, some amount is we are losing, right? So that some amount of energy is lost for the purpose of digestion and absorption of the nutrient. So that is called, so how it is lost? It is lost in the form of heat. So diet induced thermogenesis, that's what they say. So this is the extra calories. So diet induced thermogenesis is the extra calories required for the digestion and absorption of nutrients. Okay, so that is called SDA, specific dynamic action, right? Okay, so now we have some other subjects also, some questions from other subject, so which is of my interest. I'm not going to discuss all the other subject question, which is a bone forming drug they are asking. So bone forming drug means, okay, let the poll complete. So what is teriparatide? Tell me. So what is the action of teriparatide? What is teriparatide? Teriparatide. What is this drug? What is this drug? Is it a full PTH or what is this? It is a full homologue of PTH hormone. Is it a complete PTH? Right. So this teriparatide is not a full one, because if you give parathormone continuously, right, that has the bone resorption effect. Okay, so PTH continuous PTH means bone resorption, but intermittent PTH has bone formation effect. Okay, so teriparatide is not a full sequence of parathormone, so it is like you are giving intermittent PTH, so that will, the effect will be like that. So that is why it has osteoblast activity. So these are all what they are going to do. They are going to inhibit osteoclast. Okay, so they are all going to inhibit osteoclast. So can you name any other thing? So any other, there is one drug which has both osteoclastic effect on osteoclast and osteoblast. Any other drug which is having effect on both osteoclast and osteoblast? Excellent, strontium ranelate. Okay, strontium ranelate has effect on both osteoclast and osteoblast, but I think it has uh, renal side effects, right? Because of the renal side effects, the that is the limit dose limiting effect of strontium ranelate. Right. Answer this one. This is a physiology question. While passing through PCT concentration, which of the following substance will increase? So just by using logic, you can answer. Okay. Just use your logic. No need to think too much. Seshogopalan, have you used your logic? Shreya, Kritika, Nadia. So they all answered nicely, correct? Okay. KJ Kumar, excellent. Okay. So what is the normal pH of the urine? Normally pH should be what? Normally the urine pH should be? Tell me the normal pH. Is it acidic or alkaline? What? What? pH of the urine I am asking. pH of urine. Excellent, Faisal. pH of urine is always acidic. Okay. And if there is amino aciduria, glucoseuria, all this in Fanconi syndrome, when there is pan proximal tubular defect, you remember Fanconi syndrome, so there will be amino aciduria and all, right? So this is normal condition they are asking. So amino acids are all absorbed, they are precious. Glucose is absorbed, bicarbonate should be absorbed, right? So if there is defect in this, so if there is defect in this, what is going to happen? So if the bicarbonate is not absorbed, what will be the condition? Renal tubular acidosis. Is this clear? So in the blood, there will be acidosis. In the urine, the pH of the urine will be alkaline here. Is this clear, everyone? Just by using this logic, you can easily answer. Right? Okay. So based on the enzyme action, there was a graph. Okay. I'll just tell you, I'll draw the graph. Substrate, then product. So what is the energy difference between substrate and product? What will you call that? The energy difference between substrate and product, that has not been asked in the exam, just I'm asking you. The energy difference between substrate and product, I want to know whether you understood the concept. Just by memorizing the previous year entrance question, that is not really going to help you. You have to know the concept. Tell me, the energy difference between substrate and product, what will you call? Delta G. No. What? It is the delta G. Energy difference between free energy difference, right? Free energy change. Is this clear? So let me go from basics. First, this point clear? The energy. So this is the energy, free energy of substrate, free energy of product. So delta G is the difference between your energy of product and energy of substrate. So here it is negative, right? So here it is what? Negative. You can see. So it's a negative. So this is an exothermic reaction. Is this point clear? So that's what next time they can ask the questions based on this. They may not repeat the same question. I want you to understand the basics first. So substrate, product. Now, any substrate to become a product, it has to undergo a change called transition state. It has to become what? Transition state. So transition state is a momentary fleeting chemical species. It is neither substrate nor product. Okay. So what is the difference between energy? What is the energy? This one, substrate and transition state. What will you call? This we will call as delta G double dagger. That is the activation energy, EA. So without a catalyst, what's going to happen? So this is going to happen. But when enzyme is there, what enzyme is going to do? Enzyme is going to decrease the activation energy. Is this clear? So I think in the graph they mentioned is in the B. Okay, in the exam, I think it is in the B graph. Okay, Sushant, am I correct? So enzymes, they lower the activation energy. So question was based on this. So it's not like that. So some graph was going like this, na. So how can it go negative? Okay, so this actually we have a term for this. This is called thermodynamic pit. This is called what? Thermodynamic pit. Don't worry, please don't go into this thermodynamic pit and all. It's not for you. Don't worry, that won't come in the exam. Okay, just know that enzymes, they are, and one was flat. Okay, so that is also not possible. Okay, so it is like this one. I hope most of you put this answer correctly. I hope. So this, tell me. So they asked about this one, right? Blue color in anesthesia, this one. So they are fond of color coding, right? So either they ask color coding of blood collection tube. Okay, so color coding is very important. So blood collection tubes. Then they what are the other color codings they ask? This waste disposal bins, right? Especially in AIIMS. Waste disposal bins, right? Then this anesthesia one, right? So blue color what? Nitrous oxide. Okay. So first year students like Seshagopalan, Moksh Sharma, Dachayani, are you there? So don't worry about this. This and all you will study later. Right. Huh. Have you seen this? Exactly this image was given, right? Am I correct? Exactly this image was there in the exam. Salim Ghazi, don't say, yeah, you have not even given the exam. Okay. So there was an image. I hope this is the same image. I have not given the exam, so I don't know. Okay. So students was telling that this is the image they have given. So this is what? Okay. So you know about the image. Great, Salim. Great, nice. Nice. So look at this. So they have asked the effect of size and electrical charge of dextron on the filterability by glomerular capillaries. Okay. So Sushant, I just did a Google search. Okay. I just used some keywords. I could not find which book is this. Okay. I did a Google search. Then I found this image. Okay. A value of 1 indicates that substance filtered as freely as water. So this is the one value of 1. Whereas a value of 0 indicates that it is not filtered. So here, dextrons are polysaccharides that can be manufactured as neutral molecules or with negative, positive charges and with varying molecular weights. So many of you might have thought neutral will be easily filtered, right? You remember? So I think many of you might have thought that how come this like this. So you know the glomerular basement membrane. What is the charge of the glomerular basement membrane? Tell me. Glomerular basement membrane. What is the charge? Excellent. It is negatively charged. So what is the reason for that negative charge? What is the reason for negative charge? So the negative charge of glomerular basement membrane is due to, yes, glycosaminoglycans, which is lost glycosaminoglycans, and excellent other siloproteins are also there, and I think there are more reasons also, right? And in which syndrome this is lost? Nephrotic syndrome. In nephrotic syndrome, this negative charge is lost, right? So that is why, even though the size of albumin, it can actually go through that glomerular barrier, sleeve barrier. It's not going because of the negative charge. It gets repelled. Okay. So look at this. So cationic, that is positive charge. Okay, because of the negative charge, polycationic dextrons, they are like attracted. Okay. So how many of you got this question correctly in the exam? Don't worry if you missed it. Don't worry. Do not worry at all. Right. So this image was given. I think the same image was given. So this is a rare disease called trichothiodystrophy. So in hairs, there are many important things. Trichorexis nodosa. So in argininosuccinic aciduria, we learned, right? So that is a different. Okay. So Trichorexis nodosa is different. Trichorexis nodosa. Tricho means hair. Rexis means breakage. There you will see broken hair. So there you will see in argininosuccinate lyase deficiency, that is argininosuccinic aciduria. Here, what is this characteristic pattern? Tiger tail, right? Tiger tail banding. So it is like tiger tail banding will be there. So thio means sulfur, So disulfide, there will be problems in the disulfide linkages. So you have this friable hair. Okay. So brittle friable hair, you see this. Okay. So is there any other biochem question I missed? Any other biochem question I missed? So last 5 minutes we can have some discussion. So any other question I missed? Any doubts you want to ask? Okay. So we will have another special class. So very soon we will have a special class. So we will go for a special class. Let's do that. And what else? Plus members. So what are the plus course we have? From the 25th to 30th, every day 1 hour I will discuss MCQs, right? So that we will do. So 5 hours we have MCQ course. And Unacademy is going to bring lot of new features. So you will very soon you will know all about the new features. New features are coming in the next cycle. So whoever is not a plus subscriber, I will suggest you to go for plus subscription, and whoever is not a member of our Telegram group, feel free to join our Telegram group also. So you will get discussions, you can ask questions to me. Sesha, any other doubt? Moksha, any doubt? Any doubt in this? So thank you, Sushant, for giving some valuable inputs. So questions we will again double check with many students, right? So we'll release a refined PPT, refined PDF. Okay. So today's PDF, don't go with the today's PDF. Okay. We will release authentic recall. Okay. So in biochem, I think around 10 question, maybe. I think 10 questions, around 10 questions was there. 10, 12 only. Right. So it is like equal, like routine, right? It's not too high, too much. Okay, immunology. I forgot. See, in AIMS, biochemistry and immunology are same. So many times I forget. So biochem, immunology, they are same. Okay. In MBBS first year student, we in immunology as a subject we teach them in the biochemistry only. First year only they learn. So in immunology, there was one question. So that is what somatic hypermutation. Based on that, right? You remember? So the answer will be somatic hypermutation. This I have discussed in many of my previous classes also. So what is this somatic hypermutation is? See, the antibody produced in primary immune response, right? The antibody produced in primary immune response. When you compare this affinity of this one, this is called affinity maturation. This is called what? Affinity maturation. So high affinity of the antibody produced in secondary immune response. Shant, am I telling the correct question? So the question must be like the high affinity of antibodies produced during the secondary immune response or affinity maturation. So that is due to what? Somatic hypermutation. So what is this somatic hypermutation? Is that so there are certain regions called hyper variable regions which codes for the FAB, because this FAB portion of the antibody which is going to bind to the antigen, right? So here there will be a lot of deamination, so there will be purposeful mutation happening. So this mutation which is happening in this region, what they can do, it can decrease the affinity of antibody, or the affinity can be same, or it can increase the affinity. So ultimately, there is selection, right? So those which decrease the affinity, that will be ruled out. Same, they will be also ruled out. Finally, you will select only those antibodies which have high affinity. Is this clear? So this is called somatic hypermutation. We will discuss these questions. Next special class, what do you want? So I may go for immunology. Let's see. So I will design a special class soon. So all of you please join our Telegram group. So I'll take a special class, something interesting very soon. Okay guys, thanks for joining. All the very best for your results. All the very best for your NEET exam upcoming preparation. So I hope I was able to clear some of your doubts. Once again, I'm telling you, don't download this PDF. We will come up with the official authentic PDF. Okay? Right. Good luck, everyone. Bye. Sure, Mok Sharma. So we will upload in Unacademy also, in the Unacademy Telegram group also they will upload. So we will as everyone we will upload. Okay. So as not as individual, we will mix all the subject. Okay, Moks. So we will mix all the subjects and we will upload together. So we will do in Unacademy Telegram group also, in my own Telegram group also. we will upload, right? So that will be from the Unacademy side. I will contribute, and it will be a good decent PDF. Okay. Okay, bye.